Hello everyone, and welcome to another recorded lecture of BIO1 online. Today we will discuss Chapter 4, which is on the energy of life. So what is energy? Energy is the ability to do work or to bring about a change. Right? Energy is basically a way to move matter. And energy is kind of complicated to talk about, but we can break it down into two major forms. You can either have kinetic energy, which is energy of motion. So this animal over here is running because it's using energy of motion. It's contracting its leg muscles in order to produce movement. So kinetic energy is energy of motion. Potential energy is energy that has the capacity to do work. So it has the potential to do work. It can have potential to do work because of its position. For example, the water at the top of a waterfall has a lot of potential energy because if it were to fall down, right, it could move. It has a lot of capacity. It has a lot of potential to cause energy of motion. Right, that water can then move very far down the waterfall. But when it's at the top, it just has potential energy. Right? That could be illustrated up here with this girl holding this ball. Up here, the ball has potential energy. When the ball is dropped, that potential energy is transformed into kinetic energy, the energy of motion, which is allowing this ball, which is made of matter, to move. So that's what energy is. Energy can also be the energy stored in bonds. So potential energy is found in chemical bonds. What do I mean by that? When you take food to eat, let's say an apple, it has a lot of chemical energy. Right? And chemical energy is a type of potential energy because the bonds of the glucose within this apple, right, have tons of energy in it. Right? An apple has a lot of glucose, and glucose has a lot of bonds. So when I digest this apple and I can break down the glucose, every time I break one of these bonds, energy is released. So I could convert the potential energy in the chemical bonds of glucose into kinetic energy that will allow my body to use it and physically do something with that energy. So here just to illustrate, we said potential energy could be like chemical energy stored in bonds. So like this energy bar has a lot of potential energy because if you were to eat it, you can then propel yourself and do a nice workout. Right? Kinetic energy, we said, are moving objects, but light, sound, and heat are also forms of kinetic energy. Right? Light and heat and sound all involve particles or particles moving in a certain way, to simplify everything. So since particles are moving, that's considered motion. So we consider that kinetic energy. Here are just some examples of how potential and energy are converted from one another. So in this fully extended bow, right, there's a lot of potential energy. And once I release the bow, the arrow will be propelled forward. So the potential energy will be converted into kinetic energy once I release the bow. On top of a hill, you have high potential energy, but once you go down the hill, energy will be released as kinetic energy. In a compressed spring, you have tons of potential energy, but once you extend the spring, that energy is released as kinetic energy and is transferred to the ball, so the ball will move upward. So like I mentioned before, chemical bonds store potential energy. So molecules like glucose and triglycerides store energy in their bonds. And to release the energy, those bonds must be broken. And that's what happens in digestion. If the cell cannot capture the energy being released, it is lost as heat. And it turns out that heat is lost every single time potential energy is converted to kinetic energy. So just like the gasoline in your car isn't combusted to 100% efficiency. Actually, most of the gas you put in your car gets lost as heat. It doesn't actually end up giving your car more miles. So most times when energy is converted from one form to another, heat is lost to the environment. So you can pause here. And the answer is C, energy of motion. What best describes potential energy? This is D. So now we can talk about thermodynamics. And thermodynamics is the study of energy transformations. Right? It talks about how energy gets transformed or transferred from one form to another. And the first and second laws of thermodynamics describe the energy conversions required for life. We'll talk about the first law, the conservation of energy. That'll be the first law of thermodynamics. And the first law is pretty simple. It says that energy cannot be created or destroyed, but energy can be changed from one form into another. So this is, again, something you might have heard before, but this is saying is if you have a certain box of energy, you can change it to a different form, but it's going to weigh the same amount. It's going to be the same relative amount, right? You can't get more or less energy. You can just change it. So we can illustrate this transfer of energy with a couple different illustrations. Let's just use this bottom one over here. So if I want to get electrical energy, so I can power my computer. One way I can do it is I could use wood, which has potential energy, a form of potential energy in the form of chemical energy, and then I could light the wood to produce heat. Heat is a form of kinetic energy. I could then use the heat to heat up water to produce steam. So the water molecules will be moving very fast, turning into steam, which is, again, a form of kinetic energy now. And that can cause this turbine to move. So the turbine will spin because the high pressure steam will cause the turbine to continually spin and spin and spin. And if you connect that to a generator, you can convert the kinetic energy of motion into electrical energy. But what I'm trying to say is originally all of this energy you're getting comes from the wood, right? It comes from the original energy that you're burning, right? So you're not getting energy from nowhere. You're just converting it from one form into another. And that's what happens in all of living organisms, especially the circle of life, so to speak, involves energy conversions. So all energy comes from the sun. And we said the sun is a form of kinetic energy because it's consisting of photons, like little light particles that are moving in waves. And the process of photosynthesis converts the energy from sunlight into the energy of sugar. So it converts solar energy into chemical energy. And chemical energy is a form of potential energy. What animals do, like us, is we can use the potential energy of sugar, right, glucose, and we can convert it into heat energy and chemical energy of ATP. So we convert the energy in sugar. We can't just use sugar. We only can use ATP for energy in our cells. So we take the energy from sugar and we convert it into usable energy in the form of ATP. And of course, every time we convert energy from one form into another, we lose heat. So that's where you see heat being lost here and here. So like I just mentioned, in plants, solar energy is used to form carbohydrates from carbon dioxide and water. But much of that energy is lost as heat. So that's what producers could do, right? Producers produce their own energy. They do so by converting solar energy, a form of kinetic energy, into chemical energy, a form of potential energy. The second law is a little more complicated. The second law of thermodynamics says that energy cannot be changed from one form to another without a loss of usable energy. What this is basically saying is that each energy transformation moves us closer to a state where all usable forms of energy just become heat that is lost to the environment. Right? In other words, the second law of thermodynamics is stating that all energy transformations are inefficient because every reaction loses some energy to the surroundings as heat. Right? So this is what the second law is describing. So energy thus flows through the ecosystem. It comes from the sun, and then it flows from one organism to the next, and heat disperses in the environment, cannot be captured again. and converted. So all energy comes from the sun and flows through the ecosystem. It's not a cycle. So heat continually disperses in the environment over time, right? So as time progresses, more and more energy in the form of heat is dispersed in the universe, okay? And this process is irreversible, right? Cells cannot use energy that has been lost as heat. So this is what brings us to the second law. The second law explains that the universe becomes more disordered over time, right? Heat energy is disordered because it results from random molecular movements. So because heat is disordered and all energy eventually becomes heat, it follows that all energy transformations must head toward increasing disorder. And entropy is a measure of this disorder. So entropy is a term used to indicate disorder, or the way I like to think about it is energy dispersal. So what things like to do in the universe, just the laws of nature, is things like to disperse. Things like to get more disordered. So the second law of thermodynamics says that things get more and more disordered because as energy gets transformed from one form into another, heat is being lost, and heat equals disorder. So entropy increases in the universe over time. Right? And every cellular process results in an increase in entropy in the universe and a decrease of available energy in the cell. Right? So this is a very, very, very important concept. I just want to actually talk about this a little more before I go on, because organisms seem very highly organized, right? So it seems like, how do organisms continue to stay organized if they want to get more disordered over time, right? But it turns out we have a constant input of energy from the sun. So we constantly need to use the energy of the sun to maintain our organization and our order. And I'm going to explain this a little bit, but again, this is a little complicated. Entropy is one of the most complex concepts to understand in biology, I would say. Right, so things in nature go from less stable to more stable on their own. So things like to go from less stable to more stable. And we said the universe becomes more disorganized as time progresses. Right? Things are going from order to disorder. To maintain organization over time, energy is required. Right? So I'm going to give you an example of a classic example with a messy room. So here we have a very nice organized room. It's very ordered. It requires a lot of energy to keep the room this way, right? But there's very little entropy. There's very little disorder. But just over time, what's going to happen? Just again, by not doing much at all, the room will end up looking like this. Just over time, just by nature. It's going to become less ordered, right? This will require less energy to upkeep, right? You just keep on doing what you're doing, but it has a lot of entropy. And we can say that this room is a lot more stable because it's more likely to stay like this on its own. This room, in this clean, organized state, is very unstable, right? It's very unlikely for the room to just stay like this at all times, right? In order to keep this room intact, the organized effort requires constant energy input. Right? So again, what I'm trying to explain is that things like to go from less stable and more ordered to less ordered and more stable. So please take a second to reread the book when it talks about energy and entropy, but also to make sure you understand these slides. I could try to illustrate it in one other way. Let's think about children. Children have a lot of energy, right? But they're not very stable, right? This is a general trend. When things have a lot of potential energy, they're less stable. And what happens over time, just by default, the way nature works, is that things become more stable over time, but they have less energy. So as this boy grows up, he'll become a man that is more stable, right? Stays in one place more frequently, but has less energy overall. So energy is inversely related to stability. So in cellular respiration, we can use the concept of entropy. So glucose in cellular respiration is very organized, right? And what happens is it gets broken down into carbon dioxide and water and ATP, right? That's what cellular respiration does. We're getting glucose and converting it into CO2 and water and getting energy that we can use for ATP. So we're going from something that's more organized and more potential energy and less stable into something that is less organized, right? There's more disorder, right? More combinations of molecules here. This has less potential energy. Each of these molecules has less energy than this big glucose molecule. And this is more stable, right? It's more likely that carbon dioxide and water will stay as carbon dioxide and water than glucose to stay as glucose. Right? Glucose over time will just break down. That's what happens to things. Things go from more organized to less organized spontaneously. Right? That's how nature works. Another way we could apply the laws of entropy is with diffusion. And we'll talk about this in a later slide of this chapter. What happens on one side of the membrane is you might have a lot of H pluses, which are protons. So when you have an unequal distribution of H pluses, this is more organized, right? You have only one side with H plus, one side not. It's very organized. But in order to keep it this way, it requires a lot of energy, right? So it's not going to stay like this for long because things like to reach equilibrium. Ions want to always be equal on both sides of the membrane. So just what's going to happen over time with kinetic energy, there's going to be an equal distribution of hydrogen ions. Of course, this is less organized, right? You don't just have one side that's clean and one side that's H plus concentrated. But this has less potential energy and is now more stable, right? It's going to stay like this longer than like this. So again, this is how we see the relationship between energy, order, and stability. So make sure you understand those concepts. Give yourself time to really internalize it and grasp it. So read 4.1 very, very thoroughly in the book. Right? And we said that in order to create the more organized glucose from water and carbon dioxide in photosynthesis, we're going to need to input energy. So when we get these low energy molecules, CO2 and H2O, and we want to get carbohydrates like glucose, we need to input energy from the sun. Right? And the ultimate fate of all solar energy in the biosphere is to become dissipated in the universe as heat. So over time, right, all of the energy from the sun is going to be dissipated and lost in the universe as heat. So again, this raises the question is how can a cell maintain so much organization, right? Because organisms are so highly organized, it seems like they defy the second law of thermodynamics. But a living cell can function because it is a temporary container of order purchased at the cost of a constant flow of energy. In other words, if we have a constant stream of incoming energy from the sun, that will allow organisms to maintain their organization and stay alive using the information in their DNA. Right? In other words, organisms can continually increase in complexity as long as something decreases in complexity by an even greater amount. So finally, I just want to illustrate this one more time, right? Life can continue to become more ordered and complicated because the sun is constantly supplying energy to our planet, right? But the energy is accumulating, right? Entropy is accumulating in our universe, right? Because the sun is continually giving us energy that is being lost as heat. So at a cost to our keeping organized, right, there's an increase of entropy in the universe. So there's going to be an increase of heat in the universe in order for us to keep our organization. And like we said, in order to be alive, we need to keep organized. So this is how energy is crucial for our organized life. So I'd like you to watch this video on YouTube. It talks a little bit about understanding the second law of thermodynamics in a little more detail. I won't go through it now, but that link was down there. So now let's talk about chemical reactions. And in every single cell, thousands of different chemical reactions have to occur just perfectly. And a chemical reaction is simply the making of bonds or the breaking of bonds. So if you have molecules breaking bonds or forming bonds, you're doing a chemical reaction. The things that start in a reaction are called the reactants. So glucose and oxygen are the reactants in this reaction, and those form the products of carbon dioxide, water, and ATP. What reaction is this? Right? This is cellular respiration, right? This occurs in the mitochondria of eukaryotes. So glucose and oxygen are the starting materials, the reactants, and they form the products of carbon dioxide, water, and ATP. The word metabolism encompasses all the chemical reactions in a cell, including all those that build new molecules and all those reactions that break molecules. So anabolism is how we take smaller molecules and use energy in order to build those smaller molecules into larger molecules. That's anabolism. It's building things. Catabolism is the other arm of metabolism, which breaks down larger molecules into smaller molecules. And as a result, we can release energy. So metabolism is the sum of anabolism and catabolism. So if you have a very fast metabolism, does that mean you're doing more catabolic reactions or more anabolic reactions? If you have a fast metabolism. Right? If you have a fast metabolism, that means you're breaking things down faster than you can build them up. So you have more catabolism going on. So I just want to illustrate a quick concept called free energy. And free energy can be defined as the amount of usable energy to perform work after a chemical reaction has occurred. I'm not going to go into too much information into this, but free energy is abbreviated as delta G for Gibbs free energy. And you can determine delta G by taking the free energy of the products and subtracting from that the free energy of the reactants. So the free energy is the amount of usable energy. And this is going to become important when we discuss these two kinds of reactions. Okay? So we can have two kinds of reactions, either endergonic or exergonic. We'll talk about exergonic reactions first. In an exergonic reaction, we have energy being released. This gives us a negative delta G. This is negative, we got a negative delta G value, which means that the reactants have more free energy than the products. If the reactants, the starting materials, have more energy than the products, that means energy must be released. Right? If you have more starting energy, if you have more energy in the starting materials, and then your products have less energy, well, where did the energy go? It had to have gone somewhere, right? Energy can't be lost. So the energy must have been released into the environment. And that kind of reaction occurs spontaneously. So exergonic reactions happen on their own. It's almost like dropping a ball from high energy, right? If you drop a ball at high energy, it automatically goes from high to low. That's the same idea with an exergonic reaction. An exergonic reaction releases energy, and the products contain less energy than the reactants. So cellular respiration is an example of an exergonic reaction because we're releasing energy from glucose. Glucose is the starting material, right? So this is an energy diagram. So this is showing the reactants, glucose, have a lot higher energy to start with. And the end products of this reaction are lower energy, carbon dioxide and water. So if you go from high energy reactants to low energy products, there's this much energy being released. So when you go from high to low, you're going to release energy in this amount. So this is an example of digestion. In an endergonic reaction, energy is required. So that requires an input of energy, right? Endo means put into. And that means that the products contain more energy than the reactants. And of course, this will not happen spontaneously. You need to input energy, right? So building muscle, you can't just automatically build new muscle. You have to lift weights. You have to physically put in energy to convert amino acids into proteins. So that's like going uphill, right? If you're starting off with low energy reactants, but you want to build them up into a high energy product, you need to input energy. So endergonic reactions require energy. And an example of that is in photosynthesis. We're taking low energy CO2 and water, putting in tons of energy from the sun so we can make glucose. So that's an endergonic reaction. So if the products of a reaction have more free energy than the reactants, the reaction is... So the answer is, it must be endergonic, right? Because the products of the reaction have more free energy than the reactants. That means that something must have required energy. The reactants required more energy to become the higher energy product. If energy is released from a reaction, it is considered what? Pause here. And the answer is B, exergonic. Right? And remember that when that energy is released in an exergonic reaction, some is lost to the environment as heat, and entropy will increase. So now we could talk about oxidation and reduction. So electrons can carry energy, and electrons pass from one molecule to another, thus transferring energy. So oxidation means the loss of electrons, right? Or loss of energy from a molecule. Reduction is the gain of electrons, or the gain of energy. And oxidation and reduction happen simultaneously. So what we call these, we call them oxidation-reduction reactions, or redox reactions. And this is how energized electrons get transferred from one molecule to another. You can remember this by oil rig. Oxidation is loss of electrons. Reduction is gain of electrons. And it's very much like giving a gift, right? So over here, we have this woman is giving electrons to this other woman. This woman over here is being oxidized, and this woman over here is being reduced. So the molecule that receives the electrons is reduced. And you could remember reduced because if you get an electron, you're going to lower your charge, right? If I gain an electron, I'm going to have a -1 charge. So my charge is being reduced. Oxidation, right? If I lose an electron, that means I'm actually going to become more positively charged. I'm getting rid of a negative. So redox reactions can link an exergonic process with an endergonic one, right? The oxidation half is exergonic since energy is being lost, right? The energized electrons are being removed. But the reduction part is endergonic because the acceptor molecule has gained energy-rich electrons, so it now has more potential energy than it had before the reaction started. So in the production of NaCl, what is oxidized and what is reduced? So if you remember, sodium had to lose an electron, so that was oxidized, and then chlorine gained an electron, so chlorine was reduced. In an electron transport chain, we have groups of proteins that do reduction and oxidation in a sequence. So some molecules act as electron carriers. And what happens is one electron carrier can donate an electron to one protein, which can then transfer that electron to another protein, which can transfer the electron to another protein, transfer the electron to another protein. So what's happening is we're doing a series of oxidation and reduction. One molecule is losing an electron. Now this molecule is gaining an electron. then it will do the same. This will lose the electron, this will gain. This will lose, this will gain. Every single time an electron gets passed, some energy is being lost, some energy is being released. And the cell can use that energy in other reactions. So keep that in mind, because that becomes very important when we talk about photosynthesis and cellular respiration. So we can use energy donors to pass electrons from one protein to the next, and we can use some of the energy that will be released as we pass these electrons from one protein to the next. So pause here and answer this question now. So over here, we can see that molecule X is losing an electron and molecule Y is gaining an electron. So that means the answer is C. We spoke about ATP already. ATP, adenosine triphosphate, is a special nucleotide that can temporarily store energy. And this is the energy that cells can use. And the energy is stored specifically in this the second to third phosphate bond right over here. So their ATP means 3 phosphate groups, right? Triphosphate is 3 phosphates. So it's specifically this bond over here between the second and third phosphate that has tons of energy. If I were to break this bond, I could then release a lot of energy that the cell can use to power chemical reactions, right? And then ATP is normally kept low inside of the cell. It's constantly being made from ADP. So adenosine diphosphate, right, is, again, you have your adenine, your sugar, but 2 diphosphates, you have 2 phosphates. So that's like a partially charged battery. You have some energy. But when I tack on another phosphate with some energy, I can then form a fully charged battery. This is ATP. That's what we like. So what we could do is so we're saying we're adding a little bit of energy to ADP so it can keep the high energy ATP. Well, where does that energy come from? Glucose, right? It comes from the energy that we eat in us, right? So we need to eat glucose so we can convert this partially charged battery into fully charged batteries that can be used for the cell. So ATP powers the mechanical, transport, and chemical work of the cell. So, for example, motor proteins like kinesin need to use ATP in order to move. In order to transport materials in and out of the cell, sometimes we need to use energy. We said also ATP is needed to activate certain enzymes in the cell. So energy is used that way. So here's some more examples. You can pause here. ATP can actually be used also to make heat, to regulate an organism's temperature. But there's even a more bizarre way that honeybees use ATP to convert heat. And I'll show you this little video now. So honeybees have a queen of each hive, of each colony. And that queen gets too old and eventually has to be replaced by another queen. The way that happens is that all of the queen's drones end up killing her. So they kill the queen once she gets too old and then elect a new queen. And they do this by surrounding the queen bee. Here's the queen. So all these drones are going to be surrounding the queen bee who's too old, and they're going to make ATP and use their ATP and convert it into heat. So they can basically cook the queen until she's killed. So what you're going to see here is bees swarming the queen. And then again, they're using ATP to produce heat. So they're going to kill the queen. And this is called balling. They're balling the queen. So again, another use for ATP you might not have known about. Right? And we said ATP is formed during cellular respiration. Right? So cellular respiration is the series of chemical reactions that help transfer the energy from sugar into the form of energy in ATP. So chemical reactions must occur very quickly to sustain life. Right? So in our mitochondria, we have cellular respiration occur, and we have to have constant ATP production. So again, this is kind of like a chain reaction. We need to consume energy. So if we have an energy bar, we then have enough energy to make ATP. We could then use the ATP to power an endergonic reaction, like moving, right? If I want to ride a bike, I need energy. So because I had the energy bar, I was able to make ATP, which can then power my exercise, right? We need to constantly, though, eat more energy so we can constantly make more ATP. If we stop eating, right, we're not going to make any ATP and we won't be able to move anymore. So coupled reactions are very important to understand. So coupled reactions, like their name implies, are simultaneous reactions in which one provides the energy that drives the other. So what this is saying is that energy from an exergonic reaction can be captured by ADP to make ATP. And that ATP can then be used to drive an endergonic reaction. Again, this is a little tricky, but this is what I was showing before with that image of, right, how we need to get more energy to power reactions. So this is how we get unfavorable reactions to occur. Let me explain. We need to constantly input energy in order for us to build proteins, for example. We need to constantly build muscle. So where are we getting that energy from, right? We need to take amino acids and convert them into proteins. In order to do that, we need to use ATP, right? So we need to use ATP in order to convert amino acids into proteins. But then how do we get more ATP, right? Well, we need to convert glucose. When we get glucose, we can use the energy from glucose to make more ATP. So as glucose goes to its lower energy, CO2, H2O, and heat, we can make ADP into the higher form of ATP. Right? And then, like we said before, now that we have ATP, we can then make amino acids built into proteins. I hope this is making sense. I know it's a little tricky, but once you go through this image a couple times, I know it'll make a lot more sense to you. So again, what I'm trying to say is that when you have glucose, you can break that down into CO2 and H2O. And when you do that, you can then make ADP into the high-powered ATP, right? Because we're going from high-energy glucose to low-energy CO2. We could then take a low-energy ADP and turn it into a high-energy ATP, right? It's coupled reactions. Now that we have a high-energy ATP, right, if we break that down to a low-energy ADP, we could then take low-energy amino acids and convert them into the high-energy protein. Right, so this is how coupled reactions work. Right, cells couple the hydrolysis of ATP to endergonic reactions. Right, the ATP hydrolysis reaction will drive the endergonic one, which does the work to make new molecules. Right, so this is how this will work. Before I go on to enzymes, I just want to quickly illustrate how this coupling really works. Right, so a cell uses ATP as an energy source by phosphorylating another molecule. Phosphorylation just means adding another phosphate. So this phosphate group can energize another molecule, making it activated. So just by adding that extra phosphate from an ATP, it can activate another molecule. Right, and we mentioned molecules or enzymes that add a phosphate group to another one. It's called a kinase. So a kinase adds a phosphate group to another molecule, thus activating it. Another consequence of phosphorylation may be the protein changes shape. So in response to ATP phosphorylating something, that protein might change shape and allow it to do something else. Right, so it might allow that molecule changing shape requires energy. Right, so the ATP provided the energy required to change that molecule's shape. So again, this is tricky stuff. Again, it's more like a little bit of biophysics than biology. But we need to understand these basics so we can understand cellular respiration and photosynthesis in the following chapters. So now we will talk about enzymes. So, introducing enzymes. Enzymes are large organic molecules that can catalyze chemical reactions. What catalyzes is just to speed up. So enzymes are among the most important of all biological molecules because they can speed up reactions. Most enzymes are proteins made of amino acids. However, some are made of RNA, which is a nucleic acid, such as the ribosome. So the ribosome is actually an enzyme that has its catalytic function because of its RNA, not its proteins. The reactant, or the starting material in an enzymatic reaction, is called the substrate. So the substrate, or the starting material, binds to the enzyme's active site. So the active site is a location that's very specific on the enzyme that can only recognize one substrate. And at the active site, the substrate is converted into products. Right? So again, enzymes are very specific. So the substrate fits like puzzle pieces into the active site. And then once the reaction occurs, the enzyme will release the products. And the reaction does not consume the enzyme at all. So the enzyme is reusable. What enzymes do is nothing that wouldn't happen normally. It just speeds things up. For example, if you left an apple out for 100 years, it will eventually disintegrate into its smaller building blocks like carbon dioxide and water, etc. However, I don't want to wait years and years and years to digest an apple if I eat it, right? So we have enzymes like amylase that can help break down the starch contained in the apple into sugars that can then be digested into carbon dioxide and water a lot more easily. So again, enzymes don't make the impossible happen. They just speed things up. How do they speed things up? They do so by lowering the activation energy, right? The activation energy is the amount of energy required to start a reaction. So it's like a barrier to entry. So enzymes speed up reactions by lowering the activation energy required for the reaction to proceed, right? This makes it a lot more likely for it to occur. And even exergonic reactions that ultimately release energy still require an initial kickstart. So suppose my reaction, which is not an enzymatic reaction, is to get all of my frogs from the left to the right. Right? These are my reactants, and I want to get them all to the right side. Well, there's usually a huge activation energy required. There's a huge barrier. What an enzyme does, again, it's going to do the same exact thing, except it will lower that barrier. So it makes it a lot more likely that frogs will be able to jump over this barrier and become products. So again, it's the same thing. It's happening, except it's happening faster. What do we call the energy barrier that must be overcome? I'll pause here. The answer is D. The next question is, how do enzymes lower the activation energy? Right? And the answer is, enzymes can physically position substrates so that they can react more easily. So enzymes bring reactants or substrates into contact with one another, so less energy is required for the reaction to proceed. So basically, enzymes help bonds form or helps bonds break. Right? And then this is how we would see, this is an energy diagram. This is a figure from the book that shows without the enzyme, in orange, you have a huge activation energy requirement to go from reactants to products. But in purple, with an enzyme, you have a lot less of a barrier. So it'll happen a lot more quickly. So let's give an example. So sucrose is a disaccharide that has to be digested into glucose and fructose. What kind of reaction can break down sucrose? So think about what kind of reaction, what type of reaction can break this bond here? It could break down a polymer into monomers. Right? Hydrolysis, right? So water will have to come right over here in this bond in order to break sucrose into glucose and fructose. That is going to take a lot of energy, right? So what is the likelihood of a water molecule coming just here at the perfect place at the perfect time to break down this bond? Right? A lot of activation energy required. right? It might happen, it will happen over time, but it's not going to happen very easily. So we have an enzyme called sucrase, right? Enzymes end in ase. Sugars end in ose. You should write that in your notes. So sucrase is the enzyme that breaks down the sugar sucrose. And sucrase can speed up the digestion of sucrose because it can lower the activation energy by changing its shape so that a water molecule can easily do hydrolysis and break down this bond. So the enzyme sucrase can change its shape as it binds to sucrose, so water can more easily break this bond. Let me introduce this as an example. So we said sucrose is the substrate. Sucrase is the enzyme. So we start off with an empty enzyme, and of course this is a protein made of amino acids. Here's the active site. This active site can only bind sucrose, so no other sugar will fit. It's very specific. So when sucrose binds to this enzyme at the active site, this is almost forming like a workbench. So what's happening is it's almost like the protein, the enzyme itself, can position the water molecule so it can interact with this bond and do hydrolysis. So again, an enzyme doesn't actually have hands, but it can actually change its shape. So it's called a conformational change. So this enzyme changes its shape so it can position the water at this bond so sucrose can be broken down into glucose and fructose. So again, this is a hydrolytic enzyme because it's doing hydrolysis, right? It's helping hydrolysis occur and it's speeding it up. So you can pause here and the answer is A. So pause here. The answer is C. So the substrate, hydrogen peroxide, is what binds to the active site. The products would then be D, right? Oxygen and water. So enzymes, we said, could form a complex with the substrate to catalyze reactions, right? And these reactions are just breaking bonds or forming bonds. So in a degradation reaction, you can take one substrate and break it down into two products, right? That would be a catabolic reaction, like hydrolysis. In a synthesis reaction, you can take two substrates and make them into one product. That would be anabolic, or building something, right? For example, that could be a dehydration reaction, right? And for example, does sucrase do degradation or synthesis? From the last example, right? Sucrase does degradation, right? Sucrase broke down sucrose into glucose and fructose. Reactions normally occur in a sequence and follow a pathway. So what happens is it's rarely that just one enzyme converts one substrate into one product and that's the end of the story. There's usually long pathways. So what that means is there might be at least 3 different enzymes, each of which does one small step. So let's say we start up with a square and a circle. A just attaches a bond between those two. Enzyme B only knows how to add a pentagon to this product over here, right? So then B will do that. And then C only knows how to add a triangle to this product. So again, what happens is the products of one reaction become the substrates of the next, right? The products of one reaction become the substrates of the next. So sometimes you can have 6 different enzymes required to convert substrate A into product G, right? So these happen in pathways. I like to think about it as like a car assembly line. So all these robots are in a specific sequence, and each robot is programmed to only do one small thing, right? So each robot can tighten one type of bolt or something. It can only screw in one type of screw, right? But if I switch the order of these robots, you would not be able to build a functional car, right? Because this robot over here only knows how to work on a car that's been worked on already by this robot over here, right? So each robot depends on the robot prior to it. So that's how our pathways work in our bodies, right? Each enzyme relies on a different enzyme before it to do its job. So enzymes work together in these complex pathways. So because of all these pathways, it may seem like things could get a little chaotic inside of the cell. But the cells have ways to control the rates of their chemical reactions, right? If not, we would not be able to maintain homeostasis. So negative feedback is one such way that the cells control homeostasis, in that the product of a reaction slows down the production of more product. Let me explain. Suppose we have 4 enzymes that work together to convert A into a product. Well, if we already have enough product, we don't want more, right? Why use more energy to continually make more product? So in negative feedback, the product of enzyme 4 actually inhibits enzyme 1. So it feeds back, right? It feeds back into the pathway and negatively affects it. It shuts it off. So when enough product is made from enzyme 4, it tells enzyme 1 to stop. It says, okay, we have enough now. You can hold off for now. When there's no longer enough enzyme 4, then enzyme 1 will automatically be activated again, so we can have enough product be made. So that's an example of negative feedback. The opposite is called positive feedback. And in positive feedback, a process reinforces an existing condition. So this is like blood clotting. So it's a series of chemical reactions that are initiated by damage to a blood vessel, but once one clot begins to form, the reactions accelerate, which gets more clotting and more clotting and more clotting. So like when you have one product that actually promotes the action of enzyme 1 even more to get even more and more and more product. So that's when we want to get something to happen very, very fast. But positive feedback is a lot more rare than negative feedback in organisms because that can lead to disease. So we said that the substrate interacts with the enzyme very specifically. It's almost like a lock and a key, right? Where the key is the substrate that has to fit just perfectly into the enzyme, like the lock. And enzymes are a little bit like that. But enzymes can actually change their shape, right? Locks don't change their shape once you put the key in. So the enzyme changes its shape to maximize tight binding of the substrate. This is called induced fit. So when the substrate approaches the active site actually changes its shape to accommodate the substrate better. The enzyme can then physically change its shape to catalyze a reaction. And then once the products leave, that active site is restored to how it looked originally. So again, it's a little bit like a lock and a key, but it's the lock actually changes its shape. And that's called induced fit. So it's a different model of enzyme activity. So enzymes can be reused over and over and over again as long as they're substrate. And enzymes and substrate must interact before they react. So what could increase the likelihood of an enzyme bumping into a substrate? So pause here and try to think about what would increase the likelihood of an enzyme and a substrate interacting with each other. Right? If you have more substrate, right, it's more likely that it will bump into an enzyme. Or if there's more enzyme, it's more likely that it will bump into a substrate. Also, by increasing the temperature, that will increase the random Brownian motion of these molecules, which will increase their likelihood of colliding. By changing the pH, like the relative acidity or alkalinity, you can also increase the likelihood of a substrate interacting with its enzyme. All right, so enzymes are very specific to their environment. Like an enzyme has a specific preference for the environment that it works best in. So enzymes can continually work if they're in the right environment. But even in response to a slight change in pH or temperature, you then get a very defunctional enzyme. So if the salt or the pH is too high or too low, an enzyme could become denatured and stop working. Temperature is also a very important factor. So many enzymes are active within a range of temperatures, but have a special optimal temperature in which their activity is greatest. Most human enzymes work optimally around 37 degrees Celsius, which is our eternal temperature. Whereas an enzyme from bacteria that live in hot springs might function optimally at 75 degrees Celsius. So again, every enzyme is different. So what explains this very sudden decrease in activity at these higher temperatures? Why does it just like plummet automatically? Right? That's because of denaturing, right? So at a certain temperature, the amino acids can no longer form bonds with another properly to maintain their structure, right? So if you break all these hydrogen bonds at high heat, you can't have a protein structure. And if the protein doesn't have a structure, it can't have a function. So that's why at high heat, enzymes are non-functional because of denaturing. So at high heat, hydrogen bonds will break. So you can't have an intact active site at high heat because you require all these bonds to be just perfect in order for there to be the right active site. It has to be just the right puzzle piece. So if you increase the heat too much, you're not going to have the same puzzle piece shape, right? Same idea with pH. A specific range of pH is required for an enzyme to make it active. And different enzymes are different. So like in humans, we have both trypsin and pepsin are both proteases. They're both enzymes that break down proteins. But trypsin likes to work at more neutral pHs. So like in the small intestine. Pepsin only works in the stomach. So pepsin only works at very low pH, like 1.5. So sometimes enzymes have partners called cofactors. So cofactors are non-proteins that bind to the enzyme to help activate it, right? So cofactors are often oxidized or reduced during the reaction, but they're not used. So some cofactors are used to change the shape or to help with the chemical reaction. Some common cofactors are iron. Some vitamins are cofactors, like vitamin B and C are cofactors. And electron carriers, like NADP+, we'll talk about. So all of these are non-proteins, but they're added to enzymes and to help them function. So cofactors are like helpers. Right, so a lot of the vitamins you take are required as cofactors. Specifically, an organic cofactor is called a coenzyme. Iron is a cofactor for hemoglobin. So hemoglobin is a protein that can carry oxygen. And in order for oxygen to stick to hemoglobin, we need this iron. Specifically, every hemoglobin has 4 iron molecules in a heme group. And again, these ions are required for oxygen to stick. So cofactors can help activate enzymes, but other molecules can inhibit enzymes from catalyzing chemical reactions. And there are 2 types of inhibitors we'll talk about. One is called a competitive inhibitor, and that blocks the active site of an enzyme, preventing substrate from binding. So a competitive inhibitor competes with the substrate because it binds to the active site. And if you block the active site, then a substrate can't bind to it. Suppose that children are playing a game of musical chairs, and we want to inhibit that reaction from happening as a competitive inhibitor, right? That's like if all of us right now were to sit on all these chairs so the kids couldn't play anymore, right? If we're sitting down, we're competing with them so they cannot play, right? They can't play the game anymore. We're competing with them. How can I overcome competitive inhibition? The only way to overcome competitive inhibition is if you add more substrate. So basically, if you just have another 100 children, then eventually they would be able to kick us off of the chairs, and then the children would be able to play again. The same idea is if you have a competitive inhibitor, you can add more substrate that will eventually chase off the competitive inhibitor. The other kind of inhibitor is called non-competitive because it binds at a location other than the active site. or allosteric means other site. It binds to a site outside of the active site. As a result of a non-competitive inhibitor, the enzyme will change its shape and can't properly function. But again, this is not at the active site. So the substrate can't bind to the active site because it changed its shape because of a non-competitive inhibitor binded outside of the active site. In the musical chairs example, this is like breaking the legs off the chair. Right? I'm not actually dealing with the active site, which is the seat itself. I'm just going to tear off the legs of the chair so nobody can play at all. Right? Nobody can play the game. So non-competitive inhibitors change the shape of the enzyme outside of the active site. And this is a summary. You can pause here to make sure you understand. So what is the answer to this? All right, the answer is competitive inhibitor. So many drugs and medications work by altering enzyme activity. For example, aspirin relieves pain by binding to an enzyme that normally will produce pain molecules. So aspirin inhibits the production of pain molecules. Right? Antibiotics work by inhibiting enzymes found in bacteria. Right? So we could also discover and develop new medications by understanding the shape of enzymes. So suppose I know that enzyme X is implicated in breast cancer because people with breast cancer have overactive enzyme X. Well, if I understand the shape of enzyme X, right, then maybe I could design a drug, a new chemotherapy to cure this breast cancer, right? So what part of the enzyme structure do you think is most important to understand in designing one of these kinds of drugs? It'd probably be the active site, right? So if I understand the active site of an enzyme that's involved in a disease, then maybe I can work with chemists to design a chemical that will bind to that active site specifically and prevent it from working. This way I can cure cancer that way. That's how aspirin was discovered, actually. So somebody I used to work with a few years ago actually came up with the structure of this enzyme that produces pain. And was able to come up with the structure. The enzyme is called COX-2. And then again, we can help develop pain medications by understanding this enzyme that causes pain. So it's very important to understand protein structure so we can develop drugs that can help them function better. Right, and this is just showing some of the critical amino acids in an active site. So by understanding the properties of different amino acids and how they arrange themselves in the active site, perhaps we can understand how to design a molecule that can stick to the active site and prevent substrate from binding. So some poisons can irreversibly inhibit an enzyme. So cyanide, for example, is a poison that inhibits enzymes required for ATP production. Sarin gas inhibits an enzyme at the neuromuscular junction and causes paralysis. Warfarin is an enzyme, inhibits an enzyme responsible for blood clotting. So it's like a blood thinner. So all of these poisons that kill us work by just inhibiting an enzyme crucial for life. Another example that's a little outdated now is called disulfiram. It was marketed as Antabuse, and it inhibits the enzyme acetaldehyde dehydrogenase. And this is an enzyme that normally helps us break down alcohol so we don't get hungover. Well, this is an inhibitor, right? So this stops you from breaking down alcohol. So this was once used as a treatment for alcoholics. They would take this medication before they drink. So when they would drink, they would instantly vomit. They would instantly become hungover. And they would not want to drink anymore. Again, this sounds a little barbaric to me. But again, by inhibiting that enzyme, that makes you very susceptible to vomiting, right? You can't break down alcohol, so you're going to vomit right away. So the alcoholic might be less inclined to drink. So please take a second to watch this video that talks a little bit about protein folding and how we can learn more about the structure of proteins just by looking at the sequence of amino acids. This is really, really cool. This is going to be more up and coming in the future. So we spoke about inhibitors that could basically shut off a protein or shut off an enzyme. But we have two other kinds of enzymes that almost act as an on and off switch. And I mentioned these before, kinases, right? A kinase is an enzyme that uses a phosphate from ATP and attaches it to a protein. So a kinase adds a phosphate to a protein. It phosphorylates a protein. What this usually does is activates the protein, or it helps it change the protein shape. A phosphatase does the opposite. A phosphatase removes a phosphate from a protein, usually inhibiting it. So with a phosphate added, proteins could be temporarily active, or without a phosphate, temporarily shut off. So we don't need to use inhibitors every time to shut off a protein. Phosphates, using ATP as a source, really control the on and off activity of enzymes in the cell. So I'll pause here. The answer is B. The answer to this one is C. So pause here. And the answer is B. And let's do this last one for now. The answer is B again. Great. Let's watch this quick video together from the beginning.
Enzymes are proteins which catalyze metabolic reactions involving the conversion of a substrate into a product. Enzymes bind with substrates due to their complementary shape. This is called the lock and key model. Rather like a spaceship docking into a space station. In the induced fit model, the enzyme changes shape to accommodate the substrate. Enzymes enable metabolic processes to proceed rapidly at room temperature by providing an alternative reaction pathway of lower activation energy. Without enzymes, these reactions would happen too slowly to sustain life, or would require extreme conditions of pH or temperature. Carbonic anhydrase catalyzes the following reaction between carbon dioxide and water. This reaction proceeds 10 million times faster with the enzyme catalyst than without. A water molecule is coordinated to a zinc atom in the protein. A proton is removed, leaving a hydroxide ion. Carbon dioxide binds to the hydroxide ion on the zinc, and the product is released. When a similar shaped molecule competes for the active site, it reduces substrate activity. This is called competitive inhibition. Adding more substrate increases activity. In non-competitive, allosteric inhibition, a molecule binds to a remote part of the protein, preventing substrate binding by altering the shape of the protein.
So for the rest of this talk, we will discuss transport across membranes. The plasma membrane is a selectively permeable barrier, right? That means that some substances pass freely through the bilayer, but others require help from proteins. Solutes can enter and exit by different methods, depending on two factors: how much of the substance there is on either side of the membrane, and the chemical nature of the substance. How large it is it charged, is it polar? So depending on how much of the substance there is, and what the substance is itself, that will determine the method in which it can cross the membrane. What is the membrane impermeable to? Meaning, what cannot cross the membrane? Right? Water and polar molecules cannot cross very easily. They need help with other proteins. Some water can kind of sneak through, and some small polar molecules can't cut through, but for the most part, water and polar molecules are prevented from crossing. right? They need special channel proteins to get through. Ions can never cross through the membrane. They need help from special membrane carrier proteins. And large molecules are going to need to enter or exit the cell via bulk transport in vesicles. So we'll talk about all of these things. So he said charged molecules and ions cannot cross. Water usually needs help to get through, to cross the membrane through special proteins called aquaporins, but some water molecules can kind of sneak through. Non-charged molecules have no problem going through. So if you're non-charged, non-polar, and small, you can easily go in and out of the cell. Now let's talk about concentration gradients. So a concentration gradient describes a difference in concentration between two regions. So that means that a solute is more concentrated in one region than another. So in this example, we have a tea bag and a cup of hot water. The tea is more highly concentrated near the tea bag than the rest of the cup. This forms a gradient, right? There's more concentration over here than over here. So there's a direction, right? The arrow is going from high to low, right? Starts at high concentration, goes to low. After time passes, the concentration of tea will be uniform throughout the cup, and there will be no more gradient. At this point, we can say that equilibrium has been reached, right? So again, it's not that things stop moving, right? It's over time, the gradient will disappear because random molecular motion will always increase the amount of disorder. It will always just dissipate everything. So equilibrium will be reached and everything will be at its most random. So the T molecules will be as disordered as possible, right? This dissipation will occur again by random motion. So two types of transport across the membrane. It could be either passive or active. In passive transport, we're moving down a concentration gradient. We're going from high concentration to low concentration. And whenever you're going from high concentration to low concentration, you do not need energy. So we call that passive transport. When you go from high concentration, the brackets mean concentration. So high concentration to low concentration requires no energy, right? Just like pushing a ball down a hill from high to low requires no energy. You don't need energy to transport something from high concentration to low. However, if you want to move something against the concentration gradient, right? If you want to take something from where there's less of it and put to where it's more and continually put more and more and more, you're going to need to put energy. That's like pushing a ball up. So that's going to require expenditure of energy. So let's talk about passive transport first. We said passive transport is when substances move across the membrane without direct expenditure of energy. And diffusion is a type of passive transport, right? Diffusion is the spontaneous movement of a substance from a region where it is more concentrated to a region where it is less concentrated. So just like when you spray Lysol in the corner of a room, over time it will diffuse from that corner where it is highest, and then it will be evenly distributed throughout the room. When blank is reached, the solute concentration is uniform and there is no gradient. Right? Fill in the blank. The blank is equilibrium. Right? And it doesn't mean the molecules stop moving. Right? But there is no net movement in one direction. Right? So the temperature, pressure, and molecule size all makes a difference. So if it were hotter in the room, you would smell the Lysol more quickly because the molecules are moving faster. Right? And again, this is showing over time. Molecules diffuse just by random motion until equilibrium is reached. Is this dye hydrophobic or hydrophilic? And how do you know? So this must be a hydrophilic dye because it's dissolving in the water and it's evenly distributing with the water molecules. If it were hydrophobic, you would see a separate dye layer on top. Gas exchange in the lungs is another example of diffusion because when we breathe in oxygen, our lungs have a high concentration of oxygen relative to our bloodstream. So what happens is oxygen goes from where it's highest in our lungs to where it's lower in our bloodstream. And that happens automatically. Every time you breathe in air with your lungs, that oxygen can just passively diffuse into the bloodstream without any energy requirement because it's going from high to low. And oxygen gas is nonpolar and can easily cross the membrane. And only small nonpolar molecules can cross the plasma membrane passively. Like oxygen, we said, is small and nonpolar, so it can easily just cross passively by diffusion. This is not possible for large polar molecules, however. So if you have a large polar molecule, we're going to need help from a protein to get in. We'll talk about that after. Osmosis is diffusion that refers to solvent movement, to the water movement, rather than the solute. So osmosis tells us that water moves from high concentration to low concentration. So suppose this is called a U-tube, before YouTube was out, and or zooming in over here, there's a semi-permeable membrane that only allows water to pass, but not this sugar. And suppose there's a lot more sugar on the right side than the left side. What's going to happen? Right? Sugar can't go from right to left. That's what sugar's going to want to do. Sugar's going to want to go to where it's highest to lowest. But sugar can't cross this membrane. But water can. So water is going to move from where it is more concentrated, which is over here, to where it's less concentrated, over here. So that water volume will increase on the right. So now there's going to be an equal concentration. There's going to be an equal solute concentration on both sides of the membrane. That might be a little tricky, so I'd recommend pausing and making sure you understand osmosis. Right, so in this example, suppose this is called a thistle tube. I have a 10% dye in this tube, and it's sitting in a beaker with 5% dye. And this membrane on the bottom only allows water in or out. What's going to happen over time? So pause here and tell me what's going to happen. So what's going to happen over time is that water is going to go where it's more abundant in the beaker to where it's less abundant in the thistle tube. And that's going to cause an increase in volume. So what's going to happen at the end is that inside the thistle tube, there's going to be less than 10% dye. And in the beaker, there's going to be more than 5% dye. Another way to think about this, that might be a little easier, is to think about the concentration of water. So if this is 10% dye, that means it's 90% water. And if this is 5% dye, that means it's 95% water. So where is water going to move? It goes from high to low. So it's going to go from where it's 95% to where it's 90%. And that's going to raise the water volume. So osmosis can affect cell shape. In an isotonic solution, that means the solute and water concentrations are equal on both sides of the membrane. So in an isotonic solution, that's about 0.9% salt. That means the cytoplasm has the same salt concentration as the solution outside of it. So an isotonic solution would be 0.9% sodium chloride. If I were to take blood and put it in 0.9% sodium chloride, the red blood cells would be the proper shape because water would be entering the cells at the same rate as it would be leaving. If I were to place the red blood cells in a hypotonic solution, that means the solution has less salt, right? The solute on the outside is lower than on the inside of the cell. So what do you think is going to happen in that case? If I place my red blood cell into a solution that's not very salty, what's going to happen? Right? Will it swell or shrink? Well, a hypotonic solution will swell. Because what's going to happen is there's so much more water on the outside than the inside, right? It's more watery, or it's less concentrated on the outside. So the water is going to try to balance out the salty inside, and it's going to rush inside. So water is going to come inside of the cell. The cells are going to swell up and might actually burst in a hypotonic solution. So hemolysis is the bursting of red blood cells. In a hypertonic solution, that's when the concentration of the solute is higher in the solution than inside of the cell. This is like when you go into the saltwater ocean, your cells shrink a little bit because your cells have more water in them than the ocean does. So the water from your cells is trying to balance out the salt on the outside. So your blood cells shrink or crenate if they were placed in a salty solution. So a hypertonic solution is saltier than the inside of the cell, so the cells will shrink. And osmosis determines water content in plant cells. And plant cells like to keep their solute concentration higher inside than the outside, so then water will always want to enter the cells, right? So in hypotonic surroundings, the plants can be very upright, right? Plants prefer hypotonic surroundings, meaning the inside of the cells is always more salty. So by default, water will be drawn inside of the cell. If I place the plant in a very salty environment, that's going to cause water to be lost from the cells. The water is going to be taken into the salty environment, and that's going to cause the cells to shrink because all of the water contained in the vacuoles will be released to try to balance out the hypertonic surroundings. So with hypotonic surroundings, right, water is continually drawn into the vacuoles, which creates enough turgor pressure to create these nice rigid cells. So you can pause here just to review how salt balance is critical for cells. Just as a quick little fun fact, salt balance is critical for organisms, and there's really some interesting examples. So like a marine turtle cries hypertonic tears after it lays eggs to get rid of excess salt. So it needs to get rid of all the salt from the ocean once it lays on land. So it cries actually to get rid of the extra salt. And butterflies require excess salt in their body, so they can actually drink the turtle tears in order to maintain homeostasis. So you can watch this video down here if you're interested in watching that. And then you could... Okay, so hopefully you find some interest in that. It's definitely worth the time to look at that. So one other type of passive transport is called facilitated transport. It's a variation of passive transport because no energy is needed, because molecules are going down the concentration gradient. They're still going from high to low. But these molecules cannot cross the membrane on their own. So facilitated transport requires a membrane protein. Right? It requires a protein stuck in the membrane to get these polar molecules outside in or inside out. Examples are glucose and amino acids. Those are polar molecules that cannot cross the membrane on their own. So if they want to go from high concentration to low concentration, they need to bind to special carrier proteins. So in facilitated transport, polar molecules move across the membranes by carrier proteins from high concentration to low concentration. So still no energy is required because we're going from high to low. But since it's polar or large, the molecule, we need a protein. So suppose these are glucose molecules, one by one. These carrier proteins will allow the transport of the polar molecule across the membrane. And I suggest watching this video contained in the ebook right now before you move on. So finally, what if we need to transport molecules against the concentration gradient, going from low concentration to high concentration? Well, that's where we need active transport. So in active transport, a cell uses ATP for energy and a transport protein to move a substance against its concentration gradient, right? From where it's less concentrated to where it's more concentrated. Because a gradient represents a form of potential energy, right? The cell must expend energy in order to create it. And that comes from ATP. So we need ATP and carrier proteins to go from low concentration to high concentration. So for example, the cell must always have a low concentration of sodium and a high concentration of potassium relative to the outside. And these concentrations, these relative concentrations are crucial for nerve and muscle function. So one active transport system in the membranes of these cells is a protein called the sodium-potassium pump. And this protein uses ATP as an energy source to expel 3 sodiums out of the cell for every 2 potassiums it allows in. And almost half of all the ATP that we use goes to this one little protein. So this is incredibly important. And the way that this does, right, so cells must actively keep low concentrations of sodium and high concentrations of potassium. But like we know, things are going to want to balance out. Sodium is going to want to become equal on both sides of the membrane. Potassium wants to become equal. So we need to use energy to continually make sure it does not become equal, right? We need there to be this huge difference in concentration inside and outside of the cell. So what happens is ATP can bind to this protein along with 3 sodiums from the cytoplasm. And then the phosphate can change the shape of the protein, and the 3 sodiums are released to the outside. Then 2 potassiums from outside of the cell can then join in. And the protein changes shape again, releasing those 2 potassiums in. And then again, the 3 sodiums can come in with an ATP, those can be released, and then 2 potassiums in. So this has to continually happen to make sure that we have a lot of sodium outside relative to the inside and a lot of potassium inside relative to the outside. So pause here and answer this question. And the answer is C. It uses energy because it's moving solutes against their concentration gradient. So please watch this video in the ebook now. And cystic fibrosis results from defects in active transport. Because just like having a low concentration of sodium, we need an even lower concentration of chlorine in our cytoplasm. And normally we have this protein called CFTR that pumps all the chlorine outside of our cells to make sure we don't have a lot of it in it. But if you have cystic fibrosis, that means you have a mutant CFTR protein. So normally where chloride ions are moved toward the outside of the cell, a mutant CFTR protein does not move chloride ions, and that causes sticky mucus to build up on the on the outside of the cells. And that can accumulate not only in the airways of the lungs, but also in the pancreas and liver. So cystic fibrosis can have a variety of consequences. Some could be very mild, some could be very severe. So I'm going to skip that for now. And finally, we'll talk about bulk transport across the membrane. So large molecules like proteins can't even fit through a small protein channel. So they need to be transported in or out of cells using vesicles. And this is called bulk transport, since we're transporting things in bulk, lots of proteins at once. We'll talk about exocytosis. That's how cells secrete materials outside of the cell, releasing them. And endocytosis is how cells eat or internalize materials from the outside. But bulk transport requires motor proteins to carry vesicles along the cytoskeleton using energy from ATP. So if you remember, we said those big vesicles can be carried by kinesin motor proteins walking on microtubules. So microtubules, again, are like the train tracks. Kinesins are the motors that use ATP to bring along these huge vesicles that contain stuff from one side of the cell to the other. And cells can have vesicles that fuse with the plasma membrane. And once it merges with the membrane, it can release particles to the outside. That's called exocytosis. And endocytosis is the opposite. So all these substances accumulate, and then a small portion of the cell membrane buds inward to form a vesicle, which can then be surrounding all the particles. And then a different motor protein, a dynein, would transport this vesicle on microtubules to bring it closer to the inside of the cell. And there's 3 different types of endocytosis. Phagocytosis is where large solid material is taken in by endocytosis. So for example, white blood cells physically eat viruses by phagocytosis. So they capture and engulf large particles or even other host cells. The vesicle can then fuse with the lysosome, where hydrolytic enzymes can break down whatever is inside that vesicle. Pinocytosis is known as cell drinking, so to speak. It's when vesicles form around small particles, but not large particles, and that's less specific. And finally, receptor-mediated endocytosis. I'm not going to talk about as much, but that's when vesicles are coated with special receptors that are waiting for a specific type of protein to be internalized by the cell. So phagocytosis is when you take a big chunk of something or even a whole cell and engulf it. Pinocytosis is a bunch of small particles being engulfed. And receptor-mediated endocytosis is when a receptor recognizes a specific protein or molecule and then can internalize all that substance. So please watch this to review. So look over here. The solute is blank, and its transport blank require ATP. So pause here. So the answer is A, right? So it's polar. We know that because it has a protein, right? If it were non-polar, it would not need a protein. And it does not require ATP because we're going from high concentration to low concentration. So that's a good question to know. And for each of these, try to fill in the direction. Are you going from high to low? Do you need a protein or ATP? And what's an example? So I filled this out for you, but this is something important to have in your notes. All right, here's another table from the book. And so you can pause here to review. And to summarize, chapter 4, we said that metabolism consists of anabolism and catabolism, which are types of chemical reactions. Chemical reactions are catalyzed by enzymes, and the chemical reactions can be endergonic if they require energy, or exergonic if they release energy. Energy exists in two forms. It could be kinetic, energy of motion, or it could be potential energy. Concentration gradients store potential energy, like we said. We said that active transport requires ATP, which is a molecule that stores energy, and active transport can create a concentration gradient, whereas facilitated diffusion and simple diffusion dissipate concentration gradients. So that is all for chapter 4, and I'll see you next time for chapter 5.